Okay, we are going to model meiosis. So this is my counter, and I want you to imagine that this is the inside of a cell. The first thing that we have to do is find the nucleus. So here is the nucleus. It's made of DNA that is all loose. So I have some beautiful loose kale, and it's just tucked into the nucleus, but it's all unraveled, it's decondensed. And that's because your cellular machinery needs to be able to access the DNA so that you can do transcription to copy it into mRNA and then translation to make a protein. But let's pretend that this cell here is about to do meiosis. That means that this cell is going to become either a sperm or an egg. All of the DNA in this cell needs to condense and tighten up and organize itself. And so we're going to do that. In interphase, the DNA will duplicate, but first let's visualize it condensing. So I'm going to take my DNA, I'm going to condense it, I'm going to wrap it up tight, and then it will condense into a visible chromosome. A chromosome is a linear piece of DNA. It does have a centromere in the center, which is kind of pushed in. So imagine there's a pinched in center on this chromosome here. It is made of DNA tightly wound around histone proteins and organized into chunks so that you can move it around your cell and make sure that you keep the right amount of DNA in your daughter cells. This chromosome, let's say it's chromosome number 2, here's chromosome number 1, here's chromosome number 3. So I've got 3 chromosomes. They're different types because they're different sizes. And this individual that's doing meiosis is diploid. So it's going to have a chromosome 1 from mom and a chromosome 1 from dad. And it'll have 2 sets of each chromosome. So now I have 3 types of chromosomes, chromosome 1, 2, 3. And I have 6 chromosomes, 1 from mom and 1 from dad. So this individual has 2 sets of each chromosomes. It's diploid. My diploid individual is going to make sperm or egg, so I'm going to duplicate the genetic information. Each one of these is 1 chromosome. So I'm going to duplicate chromosome number 1 from dad, chromosome number 2 from dad, and chromosome number 3 from dad. When I duplicate it, my chromosomes are held together by these purple Brussels sprouts, which represent the adhesion proteins at the centromere. So here I've duplicated chromosome number 1 from mom, chromosome number 2 from mom, and chromosome number 3 from mom. So let's talk about what we have right now. I have a diploid individual that has 2 sets of chromosomes, 1 from mom, 1 from dad. So this is chromosome 1, maternal and paternal, chromosome 2, chromosome 3. You might be like, wow, everything's duplicated, so now I don't have 6 chromosomes, I have 12, and logically that totally makes sense, but it's not true. And the reason it's not true is because we count chromosomes based on the number of centromeres. So this centromere that's holding my 2 sets of DNA together is counted as 1 chromosome. So if we count the centromeres, 1, 2, 3, 4, 5, 6, we still have 6 chromosomes, even though we've duplicated the DNA. As we duplicated the DNA, we now refer to each half of your chromosome as sister chromatids. So I've got sister chromatids attached together at the centromere with adhesion proteins. These adhesion proteins keep my sister chromatids together. They are not independent, and so they are not 2 chromosomes. This is 1 sister chromatid of chromosome number 3 for mom. An overview. We've duplicated our DNA. These are called sister chromatids attached to the centromere with adhesion proteins. I'm diploid because I've got 2 sets of each chromosome, 1 from mom, 1 from dad. My chromosome sides are identical. This is an identical sister chromatid on the left and an identical sister chromatid on the right. We're now going to go into meiosis 1, and we're going to do prophase 1. In prophase 1, your homologous pairs get really close together. My homologous pairs are the sets of chromosomes of the same type from my mom and my dad. Homo means same, and ologous refers to their evolutionary origin. So my two sets of chromosome number 3 here are my homologous pairs. During prophase 1, your homologous pairs get really close together, they kind of mesh, and that's when crossing over occurs. So I'm going to show you crossing over, where these two sister chromatids of my homologous pairs are getting really close. They're going to actually smush together, and they're going to swap DNA. So we have genetic recombination occurring. Oh yeah. And that genetic recombination allows some of the alleles from my maternal DNA to get stuck to my paternal DNA. Let's pretend that one is stuck on really well. So now in crossing over, I've taken some of the alleles that were originally on my paternal and I've moved them to my maternal. And that's really cool because if we look at the sister chromatids, where these two are identical, we no longer have identical sister chromatids. So that's where genetic recombination comes from. That occurs during prophase 1. In metaphase 1, my homologous pairs line up on the metaphase plate right in the center, and my organization is totally random. So on here I have most of my paternal on the right side. That doesn't have to be true. It could be like this. I could have some paternal on the right, some on the left. Independent assortment means the way in which this chromosome 2 lined up with maternal on the right and paternal on the left, whatever, is not the same for chromosome 3. Each one organizes independently, and that's also going to lead to genetic recombination, as we'll see in a second. During anaphase 1, in meiosis 1, I keep my sister chromatids together, but I separate my homologous pairs. And now my cell would do telophase 1 and it would do cytokinesis 1. So I would divide both of these into two new cells. So this is now a cell and this is now a cell. There is a new plasma membrane between them. What changed? In our original cell, we were diploid because we had two sets of chromosome 1, maternal and paternal. In our new cells, we're now haploid. We're haploid because I don't have two sets of chromosome 1. This is just one chromosome. And we can even count the chromosomes and convince ourselves that that's what happened. So here I have 1, 2, 3 centromeres, which means I have 3 chromosomes, and our original cell had 6. So I now have half the chromosomes I need, but they're still duplicated, which is why we need to do meiosis 2, and we have to split these again. So we're going to do meiosis 2. Everyone's going to tighten up in prophase 2. In metaphase 2, you're going to line up on your metaphase plate. And then in anaphase 2, we finally break our adhesion proteins between our sister chromatids, and our sister chromatids separate. So now we have individual chromosomes, no more sister chromatids. And the same thing will happen for our other cell, which is how we end up with 4. And if we look at the results, each of these combinations is genetically different than what we started with, and they're genetically different than each other, which is pretty cool. Where did that genetic recombination come from? Some of it came from crossing over. That's how we got these different allele combinations. This is what our original looked like. And some of it came from the organization during independent assortment. So in this cell, which would then decondense into one of your gametes, so that would be like 1 sperm, 1 egg, we now have 3 maternal DNA sets and 1 paternal. In this one, we have 2 paternal, 1 maternal, and in this one, we have 1 of each, but then we have the genetic recombination that occurred during crossing over. So I'm left with 4 gametes, each different from each other and different from the parent cell. Each of these is haploid because it only has 1 set of chromosomes. Remember, our original was diploid because it had homologous chromosomes. There are no homologous chromosomes here because there's only 1 copy. And each of these will then mature. And the idea behind it is when I have a single set of sperm and egg that fertilize and become a new individual, I'll have a full set of DNA again. So that's why I had to become haploid as a sperm or an egg, so that two haploid individuals, or two haploid cells, when fertilization occurred, would give your zygote a full set of DNA. So now this cell, which was the fusion of a sperm and an egg, has a full set. It's diploid because there's two copies of each chromosome, and it can now do mitosis to grow from a zygote to a blastocyte to a fetus and then to a small person that can exist in the world. If you have any questions, let me know. I hope this helped, and I will talk to you soon.